sub-project of the project on aging concerns the development of new tools for the investigation of subcellular processes, signaling processes, and the application of these tools to studies of events happening during aging and during neurodegeneration. They are essentially visual tools which report information from images. And this has been a tradition. There is a tradition of research in this field in Professor Tullio Pozzan's lab, and he's the coordinator of this part of the work that's going on. In this poster, I have put down some examples of what is going on in the labs in this particular area. Indicators for second messengers inside cells. An example here is an indicator for cyclic AMP, which has been developed very recently, published in Cell Metabolism. It reports on AMP levels in mitochondria, a new discovery. There is cyclic AMP in mitochondria, and it is very important in the regulation of the function of these organelles. Other probes concern calcium in various subcellular compartments, and now we are developing and applying optogenetics on a large scale. Optogenetics means being able to modulate with light the function of excitable cells, such as neurons and myocytes. And there are several such cases now being employed in the labs of our institute in Padua to study phenomena such as atrial fibrillation, which is very important in aging individuals, and epilepsy, which is also, of course, predominant and increasing with age. Two examples of the application of these tools: the study of some aspects of Alzheimer's disease. To make it extremely short, Paola Pizzo and co-workers in Tullio's lab have discovered that mutated presenilin 2, which is one of the forms of familial Alzheimer's disease which has been studied, causes this mutation in this protein causes alterations of calcium signaling in cells. And the reason is that this protein acts as a gain-of-function mutant in inducing a closer apposition of the endoplasmic reticulum and mitochondria, because it turns out that it has a function in that type of process as well, besides the well-known normal one. And just again, very briefly, interactions between neurons and astrocytes, glial cells, are important in the onset of epilepsy, and epilepsy is very severe. It has a high incidence in the aged population. Optogenetics is being used, especially, the transformed interneurons expressing channel rhodopsin 2 is used to study the interaction between neurons, astrocytes, interneurons, and the control of the onset of epileptic discharges due to this interaction. It turns out that interneurons provide a negative input for pyramidal neurons, but a positive input for astrocytes, which in turn then can act to excite pyramidal neurons instead of keeping them calm as the interneurons would do normally. I think this is all I have time to say. These are just two examples. There are several other activities going on. They are all based on imaging tools. Again, this is the take-home message: new molecules to get information.